Namaste everyone, this is Dr. Pratibha K.S. from Keli Society's S. Nijlingappa College, Rajesh Nagar, Bengaluru. Today I am before you to discuss a talk on enzymes. So, what are enzymes? Enzymes are protein molecules in cells which work as biological catalysts. Okay, without enzymes, we can't survive. In our body system, we need in every single cell enzymes to perform different role, right? So here enzymes are proteins. It means those are polypeptide chain. You know, right? All enzymes are proteins, but all proteins are not enzymes. Except ribozymes, all enzymes are basically made up of proteins, that is polypeptide chain. Okay. Then what is the role of this enzyme? Enzymes speed up chemical reactions in the body but do not get used up in the process. Therefore, they can be used over and over again. Okay? During the chemical reaction, they are not going to get used up, but they are involved in doing as a catalyst, and that can the same enzyme can be used for many more time again and again. Okay, enzymes are substance that acts as a catalyst in living organisms, regulating the rate at which chemical reactions produced without itself being altered in the process. Okay, enzymes are the biological macromolecules which speed up the rate of chemical reactions without undergoing any change. They are also called biological catalyst. Okay, if in the examination, if you get question what are enzymes, you can write this. Okay, further to understand more about enzymes, we can go through this introduction. Enzymes are valuable analytical tools and offer sensitive and specific methods of quantitation for many substances. The increasing availability of highly purified enzyme preparations both in solution and in immobilized forms permits the development of a wide range of methods. Because of the difficulties in measuring the amount of enzyme in the conventional units of mass or molar concentration, the accepted unit of enzyme activity is defined in terms of reaction rate. Okay. The international unit is defined as the amount of enzyme which will result in the conversion of a one micromolecule of substrate to product in 1 minute under specified conditions. Okay, thousands of chemical reactions proceed very rapidly at a given instant within all living cells of an organism, right? Virtually all of these reactions are mediated by remarkable molecular devices called enzymes. Enzymes are central to every biological reactions or biochemical reaction and are called the catalyst of biochemical biological systems. The enzymes accelerate the chemical processes in our bodies and get the work done at a faster rate. These enzymes are organic substance that increases the rate of chemical reaction, right? Most of chemical reactions take place in the mouth, stomach, and intestine where enzymes work on the food that you put in. Other enzymes speed up nerve impulses that make your heart pump, head turn, and hand pull away from the hot stove, etc. All this will be carried out by our enzymes, which are responsible for speeding up our many activities. Okay. To conclude, enzymes are proteins that has a biological catalyst. Okay, and the catalyst, they are the catalyst that accelerate chemical reactions. The molecules upon which enzymes may act are called substrate, right? And the enzyme which convert the substrate into different molecules known as products. Almost all metabolic processes in the cell need enzyme catalysis in order to occur at rates fast enough to sustain life. Further, metabolic pathways depend upon enzymes to catalyze individual steps. The study of enzyme is also called enzymology. Metabolic processes and other chemical reactions in the cell are carried out by a set of enzymes that are necessary to sustain life. Right? Enzymes are found in all tissues and fluids of the body. Catalysis of all reactions taking place in metabolic pathway are carried out by intracellular enzymes. The enzymes in plasma membrane gum the catalysis in the cells as a response to cellular signals and enzymes in the circulatory system which regulate a clotting of blood. Most of the critical life processes are established on the functions of enzymes. The first enzyme discovered was distase, which catalyzes the conversion of starch to maltose, and that was observed in the year 1933 by two French chemists, Perrin and Furnos. However, it was not well known until 1876. Just imagine, we were not knowing about the enzymes until 1876 when Wilhelm, the distinguished German biochemist, proposed the term enzyme. Okay, so how we can conclude what are enzymes? The enzymes which are present in the living cells are biological system which is acting like a biological catalyst, right? The enzymes which are responsible to convert substrate into products, right? Enzymes which convert substrate into different variety of products. And these enzymes are basically biological catalysts which remain unchanged by chemical reactions as a catalyst. Catalyst should not change, right? Because the catalyst will be used again and again to perform many reactions. These catalysts also increase the rate of chemical reaction, right? Further, the enzymes are basically made up of proteins except ribozymes. All enzymes are proteins, but all proteins are not enzymes. Okay, now let us go through the discovery of this enzyme. French chemist Anselm was the first to discover an enzyme. Okay, that is distase in the year 1833. Further, in 1878, Wilhelm Friedrich Koene used the word enzyme to indicate the catalysis taking place in the biological systems. Isolation of enzyme system from cell-free extract of yeast was achieved in the year 1883 by Butcher in the year, okay, and later in the year 1976, James Summer first achieved the isolation and crystallization of the enzyme urease, okay, from jack bean and identified it as a protein. Okay, this is a brief history of enzymes. In the exam, if they ask for the question 10 marks, what are enzymes, and give the history and give the classification. For the history, you can write this information. Okay, further, you can also keep this in your mind. The very existence of biological catalysis was first recognized and described during the late 18th century while studying the digestion of meat by secretion of the stomach. Subsequently, Louis Pasteur concluded in 1850s that fermentation of sugar into alcohol by yeast is catalyzed by ferments. Okay, he also postulated that these ferments were inseparable from the structure of living yeast cells. Okay, and a view called vitalism that prevailed for many years. Ephraim Crane coined the term enzyme in the year 1876 to represent the ferments. The first enzyme was isolated by E. Butcher in the year 1903, for which he was rewarded Nobel Prize in the same year. The protein nature of enzyme was first discovered by James Summer in the year 1926 when he purified the enzyme urease and obtained it in the crystalline form. For this, Summer was awarded Nobel Prize in the year 1946. Okay, we can write this particular history, or you can write this history of enzymes. Okay. Now let us go through the concluding definition of the enzymes. Enzyme What how we can conclude here? An enzyme is a protein is synthesized in living cells and catalyzes or speeds up a thermodynamically possible reaction so that the rate of reaction is compatible with the biochemical processes essential for the maintenance of the cells. It is sometimes called as organic catalyst or biocatalyst. Okay, over 90% of enzymes are simple globular proteins. Many enzymes have relative molecular mass of between 10,000 and 50,000 daltons. The first enzyme discovered was amylase, which catalyzes the conversion of starch to maltose in the year 1833 by two French chemists, Pan and Pernos. However, it was not well known until 1876, okay, when Wilhelm, the distinguished German biochemist, proposed the term enzyme. We can also write the further information that enzymes are colloidal, thermolabile, made up of proteins except ribozymes and are specific. Okay, this also you can mention. I'll repeat. Enzymes are colloidal, thermolabile proteins and they are very specific in their nature. Now, again, if you want, you can write this brief information about what are enzymes. Enzymes are biological catalysts which catalyze almost all biological reactions. They are conjugated proteins. Okay, the protein part of the enzyme is called apoenzyme, while non-protein part is called cofactor. The cofactor may be a metal ion called activator or a complex organic compound called coenzyme. Okay, I want you to sit this to this particular video along with your pen and paper so that you can make a note. If I'm going very quick, you can pause this video and you can make a note. Now let us understand here systemic nomenclature. International Union of Biochemistry and Molecular Biology, in short IUBMB, adopted a scheme and suggested functional classification of enzymes and appointed an enzyme commission that is in the year 1951. As per IUBMB, enzymes are named with suffix ASE, okay, three letter with the name of the substrate and are the type of the reaction it catalyzes. Each enzyme is assigned a name and systemic four-digit enzyme code for all the enzymes later, commonly known as enzyme commission number, that is in short EC number. Okay, this is about the systemic nomenclature. Now let us go through trivial nomenclature. In the early days, the enzymes were given names by the discoverers. Okay, whoever the discoverers will be there, no, those names will be given in an arbitrary manner. Now, enzymes are commonly named by putting the suffix ASE to the name of the substrate or with the catalytic action of the enzyme. For instance, a lipase is an enzyme which catalyzes the hydrolysis of a lipid. A fatty acid synthetase is an enzyme that catalyzes the synthesis of a fatty acids. These names of the enzymes are called as trivial names. While assigning a trivial name to an enzyme, however, no systemic rules are followed. For example, here this is an enzyme and the respective substrate or reaction type. The enzyme maltase, ASE is the trivial nomenclature of the enzyme. Okay, ASE, the suffix we are added here. We have added here. Maltase, the substrate is maltose. Urease, the substrate is urea. Proteases, the substrate is proteins. Carbohydrate, carbohydrases, carbohydrates. Lipases, lipids. Hydrolases, hydrolysis reaction. Okay, deaminases, removing amines. D stands for removal. Okay, the dehydrogenases, removing of hydrogens. Okay, these are some of the examples of trivial nomenclature with suffix ending ASE. Now let us go through the general characteristics of enzymes. Enzymes are proteins in nature. They increase the velocity of the reaction without themselves undergoing any chemical change or stress. Enzymes are highly specific in their action. Enzymes have optimum pH, that is the pH at which they have maximum activity. They are very sensitive to changes in pH. Enzymes have optimum temperature, that is the temperature at which they exhibit maximum activity. The optimum temperature for the most of the enzyme range from 25 to 40 degree Celsius. The mammalian enzyme has an optimum temperature of 37 degree Celsius. Okay, enzymes are inhibited by specific molecules called inhibitors. In some other videos, I'll be discussing about these inhibitors. In a reversible reaction, the enzyme does not affect the equilibrium state of a reaction. Okay, they affect the rate of both the forward and backward reaction to the same of extent. Enzymes function by providing different pathway which requires lower activation energy than the unanalyzed reaction. They do not alter the enthalpy of the reaction. Okay, this is all about the general characteristics of enzymes. You may get questions in the examination like what are enzymes? Give the general characteristics of enzymes. Okay, you can write all this information. Now, the last one that is for your note, enthalpy, that is nothing but the sum of internal energy and the product of the pressure and volume of a thermodynamic system. In symbols, enthalpy, that is H equals the sum of the internal energy plus and the H and the product or product of the pressure P and the volume V of the system, that is H is equal to E enthalpy is equal to E energy plus pressure and volume. Okay, this is important. Kindly make a note of this one. General characteristics of enzymes. According to the International Union of Biochemists, IUB, enzymes are divided into six functional classes and are classified based on the type of reaction in which they are used to catalyze. Okay, enzymes are classified under six main headings which relate to the chemical reactions involved. Each class is then subdivided in order to clarify the other features of the reactions. The main headings are listed below. And this is also important. Usually you may get the questions like classify the enzymes based on their chemical reactions. Okay, usually you'll get this kind of questions. Kindly make a note. This is important. To brief, first one that is oxidoreductases, then transferases, hydrolases, lyases, isomerases, and ligases. Okay, whatever we have enzymes, okay, in a biological condition, all those enzymes which are put under these six categories. Okay, for instance, here oxidoreductases, enzymes that catalyze the transfer of hydrogen or oxygen atoms or electrons. Transferases, enzymes that catalyze the transfer of specific grouping. Hydrolases, enzymes that catalyze hydrolytic reactions. Lyases, enzymes that catalyze the cleaving of bonds by reactions other than hydrolysis. Isomerases, enzymes that catalyze intramolecular rearrangements. Okay, ligases, enzymes that catalyze the formation of bonds and require HEP for the reaction. Okay, important points you can underline over here. Okay, for 2 marks, if they ask, you can write this information. Further, each enzyme has been given a four-digit number by the Enzyme Commission of the International Union of Biochemistry. The first three digits relate to the reaction catalyzed by the enzyme, and the final one is required in several enzymes with different protein structures catalyze the same reaction. Okay, enzymes are commonly proteinaceous substances which are capable of catalyzing chemical reactions of biological origin without themselves undergoing any change. I'm repeatedly telling this one, make a note. Therefore, they are called biocatalysts. Enzymes are synthesized by living cells, and hence those are called biocatalysis. Okay, usually you get this kind of question. How are enzymes classified? Describe the six major classes of enzymes. Give examples for each class. Here, I'm giving the answer for 5 marks questions also and 10 marks questions also. Kindly make a note. Okay, not necessary to write entire information. You just go to that one. Whatever necessary for 5 marks and 10 marks, you can make a note. I request you all to, from this slide, you must make a note. Okay. Whenever they asked how are enzymes classified and described, first and foremost, you need to define enzymes. Here I have not written, but you need to define enzymes. After defining the enzyme, then you can go through enzymes classification. Okay, now straight away I'll be going through enzymes classification. Based on the type of reaction that enzymes catalyze, okay, all these collection of the enzymes are grouped into six classes. Okay, oxidoreductases, and you can see the suffix ase. Okay, and also based on the commission number, enzyme commission number, even all the enzymes were given 4-digit enzyme code. I'm not discussing that one here. Okay, so first one, oxidoreductases, transferases, hydrolases, lyases, isomerases, and ligases. Okay, the six classes can be easily remembered from the word OTHLIL. Okay, each letter corresponds to the first letter of the name of each class. You can keep in your mind to remember these six classes of enzyme, the classes of classification of enzymes. O stands for oxidoreductases, T stands transferases, H stands hydrolases, L stands lyases, I stands isomerases, and L stands ligases. You can keep this particular word. Okay. Let us go to the first one, oxidoreductases or oxidation reduction enzyme. Okay, these are the enzymes which catalyze the oxidation and reduction reaction by the transfer of hydrogen or electron from the substrate. Okay, we can define also as these catalyze oxidation and reduction reactions. For example, pyruvate dehydrogenase catalyzing the oxidation of pyruvate to acetyl coenzyme A. Okay. For example, other than that, lactate dehydrogenase, it catalyzes the oxidation of lactate to pyruvate in presence of NAD plus. Okay, you can keep this particular reaction in your mind. Any one example you write, that will be more than enough. Okay, that is lactate plus NAD plus, which gives us to pyruvate in presence of lactate dehydrogenase. Okay, our lactate NAD oxidoreductase, which gives us to pyruvate plus NADH plus H plus. All dehydrogenases belong to this group. For example, you can take another example here, alcohol dehydrogenase. Okay, or you can also keep in your mind glyceraldehyde 3 phosphodehydrogenase. Okay, which converts glycerol 3 phosphate to 1,3 bis phosphoglyceride. Okay, in presence of glyceraldehyde 3 phosphate, it brings the oxidation reduction reaction by NAD to NADH plus H plus. Otherwise, you can keep this particular example, lactate dehydrogenase. Okay, further, if you want more information, you can write this also. Okay, you can write this or previous slide you can write. The group of enzymes which catalyze the oxidation of one substrate with simultaneous reduction of another substrate or coenzyme. It catalyzes oxidation or reduction reactions. They catalyze the addition of oxygen, transfer of hydrogen, and transfer of electrons. Okay, we can write this particular examples also here. The same thing, lactate dehydrogenase, right? This is the lactate which gives rise to a this particular product with NADH. Okay, what happens in this particular case? This In this case, what happens here? This will be removed, right? In presence of lactate dehydrogenase, which brings the oxidation and reduction reaction. For instance, here, you can see here, the AS2, the reaction will be like this, AS2 plus B, which is going to give rise to A plus BH2. Okay, you can write either this reaction or you can also write this particular reaction. Okay, in presence of lactase, okay, in presence of lactate dehydrogenase, which is convert the lactate which is get converted into a pyruvate and NADH plus H plus. Okay, you can write this particular reaction. Further, transferases, these are enzymes which remove group other than hydrogen, that is from substrate, and transfer them to acceptors other than water. For example, ATP dehexase 6 phosphotransferases, that is hexokinase, transfer phosphate from ATP to glucose. Okay, reaction you can make out here, ATP plus D glucose, which gives a G6 phosphate plus ADP. Alanine alpha ketoglutarate aminotransferase, that is alanine, in presence of alanine transaminase, transfer amino group from alanine to alpha Kg. Okay, that is reaction here, alanine alpha Kg to pyruvate plus glutamate in presence of alanine transaminase. All kinases and transaminases belong to this particular group. Okay, they catalyze the transfer of a group from a donor to acceptor. Okay, for instance, here transaminases catalyzes the transfer of amino group from alanine to oxaloacetic acid. Okay, if you want, you can keep this particular written um, reactions here. Okay, here transferases. This class of enzymes transfer enzymes transfer one group other than hydrogen, that is from substrate to another substrate. For example, methyl, alcoholic or aldehyde, ketone, acyl, amino or sulfur or phosphate group that will be transferred. For example, here we can make out the transfer of keto group from here to here and the amino group from here to here. Okay, this is the uh, action carried out by the transferases. Here, alanine, which was okay, reacting with the alpha keto glutarate in presence of transferases, which brings pyruvate and glutamate. Okay, we can write this particular previous information, or you can also keep in your mind this reaction. Either you can write this particular information directly, okay, or you can write this information with the reaction. So this is about the transferases. Next, let us go through here hydrolases. Okay, hydrolases, these are enzymes that catalyze the hydrolytic cleavage of covalent bonds using compound of water. Okay, these hydrolyte peptide ester glycosidic and anhydrate bonds. For example, urease, that is urea amido hydrolase, catalyzes the hydrolysis of urea into carbon dioxide and ammonia. So here in presence of the urease, which converts urea, okay, into carbon dioxide and ammonia, which brings the hydrolases. That is hydrolases, that is nothing but the hydrolytic enzymes. All peptidases like pepsin, trypsin, etc. Okay, and carbohydrases, carbohydrate hydrolyzing enzymes like invertase, maltase, etc. Lipases are the examples of this particular hydrolases classes. Okay, they catalyze the reaction which involves the addition of water molecules to a bond of substrate. Okay, hydrolases are further classified into peptidases, glycosidases, esterases, many more. Okay, for example, peptidases catalyze the hydrolysis of peptide bonds. Glycosidases catalyze the hydrolysis of glycosidic bonds. Okay, this is about the hydrolases or hydrolytic enzymes. If you want, you can write this information also. Okay, hydrolyzers are hydrolytic enzymes. These enzymes catalyze the hydrolysis of carbohydrates, esters, and proteins by adding water and then breaking bonds. For example, you can keep in your mind lipases, esterases, proteases, etc. Okay, here you can see the example of triglyceride. Okay, in presence of lipase enzyme, okay, this lipase enzyme which is going to break at this particular portion, okay, and which is going to add up here this as H, okay, that is CH2OH, here also H, right, that is CH2OH. You can make a note here, right, here you can see the CH2OH, so here also H. By the addition of the water molecule hydrolysis, which is going to give a product like glycerol and fatty acid, whereas here this will add up OH, okay, and they become here also OH, and here also that is going to add OH, and they become CH3, CH2, COOH. So you are going to get your three molecules of fatty acids you are going to find here. Okay, it means which is going to give a one from one component to you are going to get here the two components. Okay, like that is glycerol, and here you could see the fatty acids in presence of lipase, which brings the hydrolysis of the product that is carried out by the hydrolytic enzymes. All digestive enzymes are hydrolases, and also the lysosomes which are filled with the hydrolytic enzymes are hydrolase the uh, you hydrolyze the whatever the component which are taken through endocytosis. Okay, and other best example is urease is a very good example of this hydrolytic enzymes. Okay, hope I made myself clear. You can write this particular information of hydrolysis in the slide 23. Okay, or you can write this information along with reaction. Okay, now let us go through the next one, lyases. These are the enzymes which remove group from substrate, leaving double bond by the mechanism other than hydrolysis. Okay, they catalyze either the removal or addition of a group of atoms from substrate without hydrolysis or reduction. For example, decarboxylases, aldolases. Okay, a malate dehydrogen malate hydrolase. Malate hydrolase, that is fumarase, catalyzes the interconversion of malate and fumarate. Malate, it get converted into fumarate, okay? And similarly, aldose catalyzes the breakdown of FDP into G3 phosphate and DHAP, okay? You can write this example for the lyases. Or you can also write for the lyases this information in short. These are enzymes that catalyzes either removal of groups from substrate by means other than hydrolysis, usually with the formation of double bonds or addition of groups across the double bonds. Okay, here you could see the addition of the OH. Okay, here is the double bond was there, and there is a addition of the hydroxyl group can be absorbed here. That is, in presence of fumarase, okay, the fumarate which is converted to malate. Next one is isomerases. These are enzymes which catalyze the interconversion of isomers like optical isomers, geometric isomers, etc. They can, or you can also write in a simplified form. They catalyze the reactions which involve changes in geometry or structure of a molecule. This class include epimerase, racemases, and tautomerase, etc. Example, glyceraldehyde 3-phosphate isomerase. Okay, that is which converts into a triose phosphate isomerase. Similarly, hexose phosphate isomerase catalyzes the conversion of G6 phosphate to of FGP. Okay, here their structure will be same and also their molecular weight also will be the same. Only change in position. Okay, they remain same in their molecular weight. Only they change in their positions. Okay, you can write this information also here. These enzymes catalyze intramolecular group transfer, that is transfer of groups within the same molecule without changing any kind of molecular weight. These enzymes can produce optical, geometrical or positional isomers of substrate. Okay, for example, epimerases, mutases, racemases, okay, cis trans isomerases you can keep in your mind. Example here, 3-phosphoglycerate. In presence of phosphoglycerate mutase, what is happening here? In this particular case, the hydroxyl group will be shifting here, okay, and this entered phosphate group is going to shift here. Okay, then the 3-phosphoglycerate will become 2-phosphoglycerate here. Next one is ligases. These are the enzymes that catalyze the linking together of two molecules coupled to the breakdown of ATP. Okay, these are also known as synthetic synthesis, or they catalyze the reactions which involve the union of two molecules by breaking of the pyrophosphate bond of ATP to ADP. And for the reaction, they need compulsory ATP molecule. Okay, example, synthetases or carboxylases. Okay, aspirate ammonia ligase, aspargine synthetase. Aspirate in presence of ammonia and ATP get converted into aspargine AMP plus inorganic pyrophosphate. Second example, acetyl-CoA carbon dioxide ligase, that is acetyl-CoA carboxylase. Okay, which is going to give rise to malonyl-CoA plus ADP with inorganic phosphate. All synthetases belong to this particular group. Okay, you can also write this information related with the ligases. These enzymes link two substrates together, usually with the simultaneous hydrolysis of ATP. In Latin, ligate, to bind. This ligases which catalyze the formation of CC or CS or CO or CN bonds by condensation reaction involving ATP. Okay, for example, you can keep in your mind ligases play a very important role in connecting the 3' hydroxyl to 5' phosphate group by forming the phosphodiester bond by utilizing ATP. Okay, here you could see the DNA strand, that is 3' hydroxyl group, plus here you could see the 5' phosphate group of the DNA strand. Okay, in presence of DNA ligase, which utilizes the ATP, which get converted to ADP plus inorganic phosphate, and which is going to give a phosphodiester bond between the 3' hydroxyl group and the 5' phosphate group. Okay, this is all about the classification of enzymes based on their chemical reactions. Okay, to summarize, you can also take a screenshot of this particular classification of enzymes and you can make a note. I have given here the 3 columns. First one is group of enzymes, what we classified, the 6 enzymes, their reaction catalyzed, and their examples. Okay, this will give you the brief information about the entire classification of the enzymes. Kindly make a note of this one. Okay, so here oxidoreductases, transfer of hydrogen and oxygen atoms or electrons from one substrate to another. Example, dehydrogenases, right? Oxidases. Then transferases, transfer of specific group, that is a phosphate or methyl, etc. from one substrate to another. Example, transaminase kinases. Hydrolases, hydrolysis of a substrate. Example, esterases are digestive enzymes. Isomerases, change of the molecular form of the substrate. Phosphohexoisomerase or fumarase. Lyases, non-hydrolytic removal of a group or addition of a group to a substrate. Decarboxylases, Haldolases, you can keep in your mind example. The last one, ligases are synthetases joining of two molecules by the formation of new bonds. Okay, this is the reaction catalyzed by that particular enzyme. Example, citric acid synthetases or DNA ligases. Further, enzymes which are grouped in six major classes have given in brief information. We can make a note of this particular explanation related with the six enzymes. Okay, in short, let me recall here. This is the concluding remarks of enzyme classification. Oxidoreductases, double-barreled name, catalyze the reduction or oxidation of a molecule. As the name suggests, oxidoreduct means which brings the oxidation or reduction. Transferases catalyze the transfer of group of atoms from one molecule to another. Example, transfer of phosphate between ATP and sugar. Hydrolases catalyze hydrolysis reaction and reverse reactions. Example, hydrolysis of an ester. Isomerases catalyze conversion of a molecule into isomer. The cis-trans conversion of malate and fumarate. Okay, Lyases reaction which break a double bond and add small molecule. Okay, that may be water or ammonia, and the reverse reaction, elimination of two form of to form a double bond, not involving hydrolysis or oxidation. Ligases, these enzymes catalyze the reactions which make bonds to join together, which means that ligate smaller molecules to make larger ones, and they use this compulsory ATP as their energy. Okay, this is all about the classification of enzymes. Okay, I've given more option here. Any one particular slide you can use to write for the classification of enzymes. Okay, further classification according to their location. Based on the location, we can classify the enzymes into intracellular enzymes and extracellular enzymes. Let us go to the first one, intracellular enzymes. These enzymes are synthesized and retained in the cell for the use of cell itself. Okay, they are found in the cytoplasm, nucleus, mitochondria, and chloroplast, and are known as intracellular enzymes. Intra means within. For example, oxidoreductases, which are involved in reduction in the mitochondria. Next one, extracellular enzymes. As the name suggests, these enzymes which are synthesized in the cell but secreted from the cell to work externally. Okay. Example, digestive enzymes which are produced by the pancreas and are not used by the cells in the pancreas but are transported to the duodenum. Okay, these are some of the examples, and this is all about the classification according to the location. This is all about the classification of the enzymes based on their chemical reactions, okay, and classifications uh, based on the location of the enzymes. Okay, in this particular unit you are going to get the questions like what are enzymes, general characteristics of enzymes, then how are enzymes are classified, describe describe the enzymes based on chemical reaction, that is six major classes of enzymes, give their examples. Okay, this is all about the enzymes. Hope you understood today's class. I Pratibha Case Signing off for today. I'll be back with some more videos. Kindly subscribe this channel and like, share this video to other needy students. Thank you, everyone.